All right, let me just turn off the slide so you guys can see a little bit better. So this lecture is based on chapter 9 in your book's textbook, but I added a couple hormones I didn't go into that much detail in, so we're just going to talk about a few different hormones that relate to not only the control of metabolism, but we're going to talk a little bit about fluid balance, which you might have learned a little bit about in environmental. So for some of you, it might be a review, but I just want to make sure that everyone is on the same page with these before we finish off the semester. So we're going to talk about the endocrine system. We're going to talk about characteristics of hormones, types, receptors, how they act on a cell. We're going to talk about the glands, so our endocrine glands, where are they located, and which glands are responsible for secreting steroid-based hormones, which glands are responsible for secreting protein or peptide or amino or amine hormones. And then we're talking about the regulation. So the regulation of these hormones during exercise and then during normal like resting activities. So we had our previous module on the nervous system, and we talked about nervous system signaling responses and how those signals are propagated usually using a type of electrical potential, so using ligand and voltage gated channels. But hormones are going to act a little bit differently. So the hormone actions are not going to be as instantaneous as our nervous system actions. So the action potentials can be almost simultaneous from the time you think of moving your arm to contract to then the contraction, but the hormone responses are going to take a little bit longer. So you might have a signal to stimulate the secretion of a hormone, but it could take anywhere from a very quick response would be like a minute or so, so a few minutes, it might be hours, or it might be days before you see a stimulus. So it's takes longer to act, but typically the actions are also going to last longer. So it's more longer acting than the nervous system responses that we talked about in the previous module. They have different functions. So control and regulation of cellular activity is a huge one. We're going to focus on metabolism, glucose regulation, and some fluid balance regulation today. And they act in different ways depending on their characteristics. So depending if they're a protein-based hormone or a steroid-based hormone, we're going to see them act slightly differently at the cell site. So here are our major endocrine glands. Let me kind of move this. There we go. Okay. So if we start up at the head, we have our hypothalamus. The hypothalamus is going to be attached to the pituitary gland. Our pituitary is in 3 different lobes technically, but we're only going to talk about the anterior and the posterior today. So as 2 different lobes that we'll discuss. Then moving down, we have our thyroid gland. The parathyroid glands sit right on top of the thyroid. Then we have our thymus, adrenal glands, which are look like little pyramids sitting on top of your kidneys. We have our kidneys, which are endocrine organs. The pancreas, adipose tissue, is an endocrine organ secreting leptin, which we don't have time to talk about today, but leptin is also an important hormone. And then we have our ovaries and our testes, so our sexual reproduction organs or glands, and those are predominantly going to be secreting our steroid-based hormones. So since the reproductive organs are not, at least most of them, not significantly going to contribute during exercise, we're not going to discuss those today. So for our steroid hormones, we're going to focus on the adrenal glands. We also see secretion of steroid-based hormones, again, like I mentioned, in the reproductive organs. But for today's lecture, we're really going to focus on the adrenals. The adrenal glands are separated into 2 major component parts. That's the medulla or the cortex. And depending on which area of that adrenal gland you're looking at, we're going to see different hormones being secreted. Steroid hormones are lipid soluble, which means that they easily pass through the cell membrane, but they are going to be hydrophobic. So they're going to repel water, just like if you tried to mix oil and water, they repel each other. Steroid hormones work in a similar manner. Because they are lipid soluble, they can pass through our phospholipid bilayer. And because of that, it's going to direct its cell action within the cell. So our steroid-based hormones are going to enter through the cell membrane, the sarcolemma, and that's where they're going to exert their action. We can see the steroid hormones have the traditional or stereotypical ring structure. Ring structures are associated with being hydrophobic, so when you see those rings, usually it means it's not going to interact with water very well. Then we have our protein or peptide-based hormones and our amino acid or amine-based hormones. The amine hormones are derived from, they're going to have nitrogen groups, but they're also derived from tyrosine. So that's how we know that those are going to be our amine hormones. If they're not based from tyrosine, then they're just going to be called a peptide or protein-based hormone. And the primary glands that's secreting those are listed here. So we have our hypothalamus, pituitary, the thyroid, adrenals as well, and our pancreas are going to be the predominant secretors of those protein or peptide-based hormones. Unlike our steroid hormones, our peptide-based hormones are going to be hydrophilic and lipophobic. So hydrophilic means that they're going to easily dissolve in water, so usually they're going to need some kind of a carrier, but they're not going to pass easily through the cell membrane, right? So they're going to be charged, they're going to repel across that phospholipid bilayer, and so they're going to exert their action on the cell membrane. So a steroid hormone is going to enter the cell, bind into an intracellular nucleus receptor. Our protein or peptide-based hormones are going to bind at the cell membrane. So they're going to bind at the cell membrane and typically use some sort of second messenger to propagate their signals. So how do hormones work and what triggers them to be secreted? So unlike maybe necessarily action potentials, or we talked about different graded potentials, we have the secretion of our hormones, which really predominantly works through a negative feedback system. The depending factors, or the deciding factors on how much is secreted, a lot of times is going to come downstream. So if we have a lot of a circulating hormone, maybe a lot of a substrate available, then we're going to see that feedback onto the endocrine gland and either increase or decrease its secretion. When we look at the plasma concentration, that fluctuation spans about minutes to hours. The overall concentration will fluctuate more across days and weeks. So again, our hormone responses are much longer acting, right? So it's not a second to second or minute by minute thing like our nervous responses. We're going to see these fluctuations, and the homeostatic control usually span longer periods of time. A good example of negative feedback, we've discussed already the glucose homeostasis in our previous lecture, but a really easy way to think about it is the thermostat in your house or in this building. And this, I think the one in this building is broken a lot of the times. You guys can let me know if you think that's true. But anyway, if it's working correctly, hypothetically, we set our thermostat at, say, 72 degrees. 72 degrees, if the temperature drops, that's going to trigger our thermostat. That's going to cause an increase in heat production, so the heater's going to kick on. That's going to cause the temperature in the room to rise. Temperature in the room rises, that's going to tell the thermostat that now our temperature has gone back to 72. That will signal the thermostat to shut off. Room will stay quiet for a while, then someone opens the door, lets the wind in, drops the temperature again, and the system restarts, right? So our body works the same way. Depending on what gland you're looking at, they are constantly having perfusion of blood. And within that perfusion of blood, they're going to have different factors that might influence the secretion. And those factors will vary depending on what hormone you're looking at. So it could be glucose, it could be insulin, it could be how many free fatty acids you have. It could be if you're feeling stressed, it could be how much sodium you have, how much fluid you have. It really depends on what hormone you're interested in to decide which factor is going to cause that stimulus for secretion. I put a little asterisk next to negative feedback because if you did look in your Brooks textbook, then you already have a spoiler alert for today's lecture, and that's glucose homeostasis during exercise may not always utilize a negative feedback system. So there's a feed forward mechanism to glucose maintenance during exercise, and we'll talk about that as we continue in our lecture today. Here's the classic example though, and this is straight from our previous one of our previous lectures. When you look at the glucose concentration in the extracellular fluid, we can see that depending on what that concentration does, we're going to see that negative feedback occur either to upregulate or downregulate the amount of either insulin or glucagon that we have. And depending on which hormone that you're either upregulating or downregulating, that will determine what happens to your blood glucose. So just to go through this flow chart quickly, we have an increase. If we have an increase in the concentration of our extracellular fluid volume of glucose, that's going to stimulate the pancreatic fat or beta cells, the islets of Langerhans in our pancreas. That's going to secrete insulin. Insulin will then work to decrease blood glucose, right? So insulin's trying to store glucose. It's also going to help store fat, and it's going to help with protein synthesis. So if insulin's being secreted, that means you have plenty of energy. You're not in need of energy, so you're going to store it in whatever form that you find it in. So once we have our insulin secreted, it's going to start to decrease the amount of glucose in our blood because it's pulling it into the cells. That will then normalize our blood glucose, and that will be a negative feedback on our pancreatic beta cells, thereby decreasing insulin secretion. On the other hand, if we're decreasing, if we have a lower value of blood glucose concentration in our extracellular fluid, that will stimulate the alpha cells in the pancreas. Alpha cells are going to release glucagon. Glucagon is going to affect the liver. It's going to start breaking down stored glycogen in the liver and in the muscle. It's going to trigger gluconeogenesis, so the creation of new glucose or glucose precursors. And then those will all be released into the extracellular fluid, thereby increasing blood glucose concentration. And that would be a negative feedback on the alpha cells. So this is the traditional feedback system for glucose, but as we'll learn a little bit later, that there is a feed-forward mechanism that is occurring typically during exercise, especially moderate to vigorous exercise. We will see a feed-forward mechanism kind of come into play before this even begins. So the actions of these hormones, like I mentioned, are in flux, and a big determinant is the plasma membrane receptors, right? So the plasma membrane receptors can either be upregulated or downregulated, depending on those different mediating factors for that particular hormone. So circulating substrates, what's going on with your blood concentrations of ions, enzymes, different things can affect this, but it's not necessarily always the concentration of the hormone that's causing its regulation, but it can also be these receptors. So one classic example is diabetes, right? So in type 2 diabetes, then the individual has typically no problem secreting or producing insulin, but they have a receptor problem. So they typically have desensitization of those receptors, so they're not stimulated to insulin as much as someone who doesn't have that perpetual circulation of glucose and insulin, or they have an issue downstream. So because insulin is a protein-based hormone, it's going to act at the cell membrane. That means it's using a second messenger, right? So if we have a desensitization in the second messenger cascade, that can also reduce our ability to pull in glucose associated with that insulin receptor. So downregulation and upregulation are the predominant ways if you're going to regulate the receptor action of the hormone. Downregulation is also known as desensitization. So for example, if you have a lot of, say, insulin, that's going to cause a desensitization of these receptors, and they either be less sensitive or they actually might come back into the cell. So they might downregulate and leave the cell surface. Upregulation is also known as sensitization. So this is the opposite, where you have a trigger. So you have a stimulus response. So say you have glucose that just appears, you have insulin that also is secreted in response to that glucose, and that will trigger the upregulation of cell membrane receptors for insulin. So in this case, this is obviously a positive response, and this is not, this response is not associated with perpetual or habitually high levels of blood glucose or insulin. So this is assuming a normal response where you're getting some sort of a trigger and that stimulus is appearing, but there's no habituation associated with it. We've already learned about our lock and key models. So we talked about this with enzymes, but it plays true with hormones as well. So the hormones are going to work with the lock and key model. There's specific receptor sites that will only match up with a specific key. In this case, it's a hormone. So hormones have action sites at specific organs or locations within the body. Because if you remember back to our endocrine schematic where we looked at all the different glands, if you have a hormone being released from your posterior or anterior pituitary, but it has to go and then act on your kidney, it has to travel through all the blood, and the blood is perfusing all the other organs. But we only want it to act where the trigger would be beneficial. So if it's traveling through your bloodstream, you only want to have receptors at that specific target site. So we do see this very specifically with hormones because they are circulating widely within our systemic blood circulation, and a lot of the target sites are far away. So when we talked about nervous system control, those synapses are right on top of each other. So it's cell-to-cell signaling, neuron-to-neuron signaling. But our hormone responses are much further away, right? So we need to have this system in place to ensure that they're activating on those sites that we want them to act on. One funny example in my one of my physiology classes I took at Nebraska is there was he was a good guy, but kind of a know-it-all student. You know, he was always kind of ahead of the professor and you know, asking questions even though he kind of knew what the response was, and always wanted to get the 100 or 110% on his labs. And so we were doing our endocrine lab, and we were looking at the hormone oxytocin. So oxytocin causes contractions specifically on the uterus. It's also involved in like a mating response and trusting responses, but we were looking at how it affected different tissues. So we had an array of different hormones, an array of tissue samples, and we were supposed to put the hormone within our tissue sample and see how it reacted and try to identify which hormone we were working with. So we came up to oxytocin, and the student kept harassing the professor. Professor, come, you know, this must be wrong. You must have given me the wrong tissue. Like I'm using oxytocin, but I keep putting more and more in, and nothing is happening to this tissue. Like nothing's happening to it. And the professor is like, okay, well, you know, let's think about this for a little bit. Let me go check my samples, and because the student was often right. So we went to check the sample. Said, no, you know, this is the correct tissue sample. You know, if you think that's oxytocin you're putting in a sample, then you just have to ask yourself, well, what does oxytocin do? And he's like, well, it contracts. I'm putting it on this tissue and it's not contracting. And the sample was from the stomach, a stomach tissue, right? So the stomach tissue. And the student's like, it's not contracting, kept pouring more solution on. Like it should be oversaturated. I'm not seeing any tissue contraction. Like what's going on? So a couple of us were looking over and we're like, well, it's stomach tissue, right? It's not uterine tissue. So we're not going to see any response because there's no receptors. And the student just turned like red in the face. Like he was so upset that he poured his whole sample over this tissue and was so upset that he couldn't see a response, but he didn't think about where the receptors were for this hormone, right? So if you're pouring the solution onto a tissue that doesn't have any receptor sites for that hormone, you're not going to see anything. It's not going to react at all. Just like if you try to get a key into a door that, you know, doesn't fit, you're either going to break the key, but most likely you can't even get it in to open it. And so we see the same thing throughout our body. And the reason for that is, can you think of a reason why you wouldn't want to have oxytocin affect every tissue in your body? So if oxytocin causes contractions, and it's typically uterine contractions to help with childbirth, would we want that to act on our stomach? Probably not, right? That'd probably be a bad thing. And so these hormones have specific actions, but they're not always gonna get along super great with every other area in our body. And so that's why we have these specific target and lock and key tissues. And that's just I think I'll remember that for the rest of my life because that kid was crazy. But that's just a really a great example of this lock and key model. So let's take a look at how these hormones are going to act. So we have our steroid hormone. Like I mentioned, steroid hormones are lipid soluble, so they can cross that phospholipid bilayer pretty easily. So that little half circle is our bilayer there. We have the hormone in red that's going through the bilayer, and then we have a nuclear receptor that it's going to bind to within the cell. So once it binds to the receptor, then it's going to start some DNA transcription. It's going to create mRNA, and then mRNA can then initiate any cellular building processes. So one steroid-based hormone, which we're not going to talk about today, but testosterone is said to be an anabolic hormone. So it had originally been thought of contributing to muscle protein synthesis response a little bit. and any other gene transcription that occurs within the cell site would go by this typical response with a steroid-based hormone. Now in your lecture, I've included a little animation, but in previous videos, YouTube has flagged me for showing video clips during this class. So if you want to look at it, then you can go ahead on, after class and take a look at the animation. You should have the link on your slide. But just to kind of go through it again, we have our steroid hormone entering the cell. Then we're going to have that hormone bind to that specific receptor in the cytoplasm or at the nucleus, and that's going to cause or activate that cell's DNA transcription, forming mRNA. And mRNA is going to leave the nucleus, and then that's where it's going to direct whatever protein synthesis might need to occur. And again, that's going to vary based on whatever cell or whatever hormone you're looking at. If we're talking about non-steroid-based hormones, these would be our peptide or amine hormones. We have, again, our phospholipid bilayer. Because we have our amine hormones are going to be charged, they're going to repel, so they're not going to easily pass through that phospholipid bilayer. Because of that, we need to utilize a second messenger system. So we're going to see a second messenger system come into play with any of these protein-based hormones, and that's going to cause what we call a signal cascade using second messengers. So really common ones are cyclic AMP, it's like one of the most common second messengers. We also have cyclic GMP, IP3, and DAG. Those are some additional second messengers. And once that initial receptor located on the cell membrane is triggered, then we're going to have the activation of that second messenger, and then we see the cascade that will then trigger any mRNA or synthesis that has to occur within the cell. So if you think about insulin, that's going to work through this mechanism. So insulin is a surface or a protein-based receptor or hormone. It's going to utilize cyclic AMP. That protein cascade is what will then trigger GLUT4, which is our glucose transporting hormone. This signal cascade will then trigger GLUT4 to come to the cell surface and help pull glucose in. So that triggering of GLUT4 would result from that cyclic GM or AMP activation. So same thing again for this. I have a video animation associated with this that you can look at when you're reviewing the materials. But again, you can see our steroid or non-steroid-based hormone binding at the cell surface. This is going to activate adenylate cyclase, which will then activate cyclic AMP. Cyclic AMP will then work. In this case, it's going into the mitochondria. Then we're going to activate those protein kinase, those enzymes to help cause cellular change. So it's still going to end up in the cytoplasm and propagating cellular change, protein kinase, and protein synthesis, but it's not going within the nucleus or the nuclear, there's no nuclear receptors for a non-steroid-based hormone. The third class of hormones, which we don't have time to get into a ton of detail with these, but they're called kind of pseudo hormones, they're prostaglandins. Prostaglandins are going to be locally acting, and they're really involved in inflammatory responses. So if you're looking at soreness or inflammation, you're usually, or even there's some relationship between the prostaglandins and headaches or migraines. You're typically going to see these at play. They're derived from arachidonic acid, and again, any kind of swelling or vasodilation response or activation of those type 4 pain receptors, prostaglandins are usually involved with that. Okay, so now we understand some of the basic categories of hormones, general ways in which they act. Y'all are getting out of learning the specific different types of cellular and plasma membrane receptor pathways, which is I guess good for now. But the endocrine system is really, really exciting. So if you have a chance to take a more in-depth class, I encourage you to do that. But we're going to cover these 4 glands, at least for now, talking about metabolic regulation. We're just going to kind of go from a heads down approach. So we're going to start at the top and kind of work our way down. So we're going to start with the anterior pituitary. If you remember back to our initial photo, the pituitary gland sits right underneath the hypothalamus. And because of that, a lot of the pituitary hormones are triggered by hypothalamic releasing factors. So the hypothalamus will typically release a hormone that will act directly on the pituitary, either the anterior or posterior, and that will cause the secretion of that anterior or posterior pituitary hormone. In this case, we're talking about growth hormone first. Growth hormone is from the anterior pituitary, and growth hormone releasing hormone from the hypothalamus is what's going to initially trigger that response. Growth hormone is involved in kind of any general muscle or cell growth, and it's going to act on a variety of different cells. With exercise, we're going to see an increase in general growth hormone secretion. So with any kind of moderate to vigorous exercise, we're going to see an increase in the secretion of growth hormone from the anterior pituitary. And again, it can act on bone. Any soft tissue or bone is where we're going to see our growth hormone act on. What we also see is that we're going to see some increase in fat metabolism, so an increase in free fatty acid circulation. With too much growth hormone, we could see a lot of people who have too much growth hormone can have type 2 diabetes. One case study, which if you had me for 121, we talk about Andre the Giant. So giganticism is related to an oversecretion of growth hormone, either because there's an issue with the anterior pituitary or at the hypothalamus, but that can lead to enlarged organs, enlarged bones, muscle hypertrophy, which skeletal muscle is okay, heart tissue not so okay. So he ended up passing away from heart complications because his ventricles hypertrophy so much that he had very, very little blood ejected, and he's a huge guy. So that is one issue that could occur if you're oversecreting or growth hormone. But in a normal, healthy individual, you're really not going to be at risk of that. And if you exercise, it's typically going to increase that. So it's going to increase the anabolic response on muscle and bone. Zach?
What if we make Andre the Giant engage in high-intensity anaerobic exercise to promote eccentric hypertrophy?
He was a wrestler.
He was a wrestler, but
so he did exercise.
Anaerobic.
So there is a treatment. So, but this is secreted with both anaerobic exercise. Growth hormone is, and you can have it treated, but he basically wasn't told, didn't go to the doctor soon enough, and when he was told, he didn't really want to do it.
You ever seen Princess Bride?
I did. It is a good movie. Have you seen the Andre the Giant documentary?
I have not.
It's a really good movie. It's on, I think it's on HBO. Yeah. But they did a pretty good documentary.
There's a documentary about Andre the Giant?
Yeah. You got to see it.
Princess Bride?
I have. Oh my gosh. This is going to be a good recording. So basically that's growth hormone. In terms of like how it relates to exercise and metabolism, it's just going to increase and help support anabolic responses. But it's not going to significantly really affect metabolism, except for a slight increase in the availability of free fatty acids to use for energy. And it does increase glucose availability a little bit as well. And that's why if you oversecrete it, you could potentially get type 2 diabetes. So anyway, moving downstream to the thyroid gland. So we have our thyroid gland. You can see it right here. One thing about the thyroid is it has smaller glands attached to it, the parathyroid gland. We're not really going to talk about the parathyroid gland too much today, but I do want to mention that is involved in calcium homeostasis. So with bone regulation and the parathyroid gland secrete parathyroid hormone, and that relates to your ability to resorb calcium from your bone. So if you had originally, and this is kind of an off-topic segue, but when they found that people had thyroid disorders, so hyper or hypothyroidism, they would take the whole thyroid gland. But they didn't realize that there were these secondary glands sitting right on top of the thyroid gland that are integral in your minute-to-minute and day-to-day regulation of calcium. So people their thyroid gland was removed, they would also inadvertently take the parathyroid glands, and that would result in significant health problems related to disruption in calcium homeostasis. So like rickets, other bone diseases, osteoporosis at very young ages, and muscular dystrophy also, there's some disorders associated with muscle contraction because they didn't have enough calcium circulating. So once they figured that out, they've gotten a little bit better when they've done thyroid surgeries to at least leave part of the gland there to allow for at least one or two of the parathyroid glands to remain and be functioning. Aside from the parathyroid gland, the thyroid itself is going to secrete two major types of hormones, so T3 and T4. Much more T4 is secreted than T3. T3 is typically going to be converted from T4, but the thyroid does secrete a very small amount of T3. The T3 is what is actually going to act on these target tissues, but again, most of what is being secreted is T4, and then eventually it's converted before its action target site. When we have the secretion of these thyroid hormone, we're going to see an increase in general metabolic rate. So your basal or resting metabolic rate will increase as a result of secretion of T3 and T4, an increase in protein synthesis, an increase in the density and size of mitochondria. So we talked about mitochondrial adaptations. Part of the stimulus for that is a secretion of T3 and T4. Increase in glucose uptake, so they're related to the translocation of our GLUT transporters, so GLUT1 and GLUT4. Glycolysis and gluconeogenesis, and then they're going to mobilize free fatty acids. So they're involved in metabolism and using energy, right? So they're going to allow you to have energy available to use. They're going to help you create some energy and help you utilize that energy via the mitochondria. Now, how are they related to exercise? So we have the anterior pituitary that is involved in secreting thyroid stimulating hormone. So not only does it secrete growth hormone, but it secretes TSH as well. TSH is going to act on the thyroid gland directly to secrete T3 and T4. T3 and T4 are going to increase, again, protein metabolism, metabolic rate, glycolysis, gluconeogenesis, and as a function of exercise, that is a positive feedback on TSH. So with exercise, you're going to secrete more TSH from the anterior pituitary. More TSH is going to lead to a greater secretion of T3 and T4, and that will, again, be involved in the increasing of these downstream mechanisms. Do you have a question, Cass?
Yeah, if someone's like, would you say that aerobic or anaerobic contributes to that more so than the other?
For this response, it's categorized as moderate to vigorous intensity, but it can be either aerobic or anaerobic. It's more based on your heart rate response. So a lot of these exercise related functions, a lot of it comes from aerobic exercise. So a lot of this research is done aerobically based, but there is research available looking at anaerobic or resistance training, but it's only when they monitor heart rate. So you have to have like a heart rate monitor and monitor what percent of heart rate max or heart rate reserve they're working at to try to equate those responses. But it happens for both. So let's move down. So let's move down past the thyroid gland. Now we're at the kidneys. And again, sitting right on top of the kidneys, we have our adrenal glands. So first we're going to talk about the adrenal medulla. So if you cut open those little adrenal glands, the little pyramid, little fatty looking pyramids on top of the kidneys, the center portion is the medulla, and these are responsible for secreting our catecholamines, or our fight or flight hormones. Specifically, epinephrine and norepinephrine. The medulla is going to secrete about 5 times more norepinephrine than epinephrine, but they both work in a similar manner. What we find is that when we increase exercise, it's going to increase our sympathetic nervous system response, and that will increase the stimulus on that adrenal medulla to increase the secretion of catecholamines. This is in part related to that feed-forward mechanism of glucose maintenance during exercise, and making glucose available during exercise is the action of these catecholamines, and that they're triggered from the sympathetic nervous system response from exercise. But you can also have secretion of catecholamines even if you're not exercising. So if you're stressed or if you're scared, or basically an increase in general arousal can increase the secretion of these catecholamines. So it's not necessarily always going to be with exercise. When you do have these released, then we're going to see increases in things associated with fight or flight. So increases in heart rate, increases in heart contractility, so more blood pump per beat. We're going to see increases in availability of substrates, so more glucose, more free fatty acids. If because if we're getting ready to run away or to fight somebody, we need fuel to do that, right? So fuel to fuel our muscles. And it's going to increase blood flow as well to allow for these substrates to go to the working muscles. Or in the case of glucose, because glucose is shunted towards active muscle, this will provide the avenue to do that.
Interesting.
Yeah.
Would you say that anxiety would be led by overabundance of release of norepinephrine, and that's why they give like those antidepressants for like norepinephrine reuptake inhibitors?
In part, but not 100%, because a lot of the catecholamine response is short-term. So it's a short-term response. Cortisol is more involved in like a long-term stress and anxiety. But yeah, so there is the catecholamine response is very fast acting, but it's also very fast to go away. So it doesn't have as long lasting effects, whereas cortisol takes longer to get up and running, I guess you could say, but the effects are much longer lasting. That's a good segue, because now we're talking about cortisol. So a part of the same adrenal gland, and we'll kind of go back a second here. So let's take a look at the adrenal gland again. And so we have our medulla here in the center, and on the outer portion we have the cortex. And if we were to pull off a little cross-section of that cortex, that's what we're looking at now. Okay, so now we have a little cross-section of the cortex, and the cortex is in 3 distinct layers. The second layer is the zona fasciculata, and that is in charge of secreting glucocorticoids. The most predominant one, and the only one actually we're going to talk about today, is cortisol. So cortisol is secreted from the middle layer of the adrenal cortex. Cortisol is going to be a stress-based hormone, typically longer lasting effects. Small exposure to cortisol is not a bad thing. Perpetual or habituated exposure to cortisol could be bad. So like habitual exposure is associated with overtraining syndrome, or like severe stress, anxiety, long-term. Then we're going to see long-lasting disruptions in our glucose, our fat, and our protein homeostasis. In the short term, it's not a bad thing though, because if you are stressed, so for example, exercise, like maybe you're doing a marathon, right? So it's not a couple minutes. Let's say you're not like the Berlin marathon runner. So you're gonna do at least a few hours that you're running, right? So then we're actually gonna benefit from that free fatty acid mobilization. We are gonna have some protein catabolism, because we might use a small % of amino acids for energy, but it's not long-term, so we don't have to worry about severe muscle atrophy or muscle wasting. And then it does work a little bit as an anti-inflammatory, anti-immune, which short-term is not a horrible thing, but in the long term, that can obviously have a negative consequence.
So is this what they're basing all that heart rate variability training on now? Like,
cortisol?
Yeah.
I haven't read too much about that.
I
So.
haven't either. I just, like, when you briefly read about it,
Yeah.
like hormones. Like,
Yeah, there is. So one of the ways that you can diagnose overtraining syndrome is monitoring changes in max, but specifically resting heart rate. So if you're monitoring, it's really hard to diagnose, because it's different in everybody. But changes in resting heart rate or a more elevated resting heart rate response is associated with overtraining syndrome. So potentially, but usually if you're diagnosing with someone, they do, after they get the initial, like, I think you might have this, and they're doing blood tests, and they're probably going to check for cortisol as well. Because that's just a commonly tested for stress hormone. So cortisol is a steroid-based hormone. So it's steroid-based, so is it going to act at the cell membrane, or is it going to act within the cell? I just think steroid-based hormone, is it going to within the cell, right? Because steroid hormone's lipid soluble, so it's going to cross right through the plasma membrane, right into the cell nucleus. So we're going to see, again, in a short term, not a horrible thing. Long term, they can be bad, and hopefully you can see why. It's going to stimulate amino acid release from muscle. Short term, that's okay, because we might need energy. Long term, that's going to be muscle wasting and not a great thing. It's going to stimulate the production of glucose from the liver, which is very good in the short term. It's not bad in the long term either, unless you don't need glucose, right? So if you don't need glucose for energy, if you're just kind of sitting around just really stressed out, then that can increase in the likelihood that you're going to have desensitization of those insulin receptors, because you're going to have hypersecretion of insulin because you have too much glucose circulating. We're also going to see mobilization of free fatty acids. And so some triggers, physical or emotional stress. So physical stress could be exercise, a traumatic event, emotional, and then also declining blood glucose levels. So if you have less energy overall, then we'll see a stimulus for cortisol, and this could be more of a short term. So if you need energy, again, it's not going to help you if you need energy immediately, but you know, if you're still going to be exercising or hiking or whatever you're doing within an hour or so, then it can help you out. So once cortisol, or if it's going to be triggered, so let's think about the short-term version. If we have a decrease in blood glucose, that's going to release one of our releasing hormones. So we have corticotropin releasing hormone, that's CRH. So corticotropin releasing hormone is going to come from the hypothalamus. Corticotropin releasing hormone is going to act then on the anterior pituitary. The anterior pituitary is going to release, it's going to adrenocorticotropin hormone. So adrenocorticotropin hormone. So corticotropin releasing hormone will then release adrenocorticotropin hormone. That will act on your adrenal cortex, and that will eventually secrete cortisol. So we're starting up here in the hypothalamus, right? Hypothalamus is attached to the pituitary. In this case, you can see maybe the two different colors here because we have different forms of connections between the hypothalamus and our pituitary. So the anterior pituitary is connected to the hypothalamus via a blood flow system. The posterior is through neural factors. So it's not going to be blood flow. Blood flow is going to be a little bit faster. So we typically are going to see those releasing factors, CRH and ACTH, be released a little bit faster than we'd see things from the posterior pituitary. Posterior pituitary is oxytocin and ADH. And we'll talk about ADH a little bit later. But once we have cortisol or ACTH released, that has to travel from the anterior pituitary down to the adrenals on top of your kidneys, right? So it has to go through your blood circulation, find that lock and key model, attach, and then you're going to see cortisol being released. Okay, so let's move over from the adrenals now, from the kidneys, and we'll talk about our pancreas. So we've talked about insulin and glucagon, at least insulin, quite a bit already this semester, so I'm not going to spend crazy amount of time going over its general functions because hopefully everyone now knows that it's working to lower blood sugar, right? It's lowering blood sugar because you have plenty of blood sugar. You have an abundance, right? So insulin is going to be secreted when you have plenty of energy to spare. Insulin is going to be secreted again from the beta cells, the islets of Langerhans within the pancreas. So once you have that high level of extracellular blood glucose, we're going to trigger those alpha or those beta cells to secrete insulin. That's going to work to increase glucose transport, synthesis of glycogen, synthesis of protein, synthesis of fat. Plenty of energy, you know, you're not starving, you're good, you're in a state of plenty, so you can store some for later. So that's what the insulin is going to help you do. It's going to decrease gluconeogenesis because you don't need to create new energy because you have plenty. So why are you going to start breaking down protein or other intermediates if you have plenty to spare? Glucagon works in opposition, so it's known as an antagonist to insulin. It's going to work in opposition because it's going to be secreted when you don't have enough blood glucose. So glucagon is going to be secreted from the alpha cells, and it's going to act to increase the production of glucose from non-glucose sources. So we call these glucosyl precursors, so like lactate, pyruvate. That's what glucagon is going to start to trigger to be used for energy, and then the breakdown of liver and muscle glycogen as well. We also see that glucagon is in part triggered by our catecholamines. So epinephrine, norepinephrine can trigger the glucagon response. Why might we see glucagon secretion be triggered by those catecholamines? What do the catecholamines tell us?
Increase sympathetic nervous drives, so you want to have energy.
Yeah, right. You're fighting or you're flighting, right? So you're going to fight somebody or run away. So you need energy, right? So you want to try to mobilize that energy. So glucagon is the hormone that's going to help. In addition to the catecholamines, glucagon is a hormone that's going to help you mobilize. So talking about our regulation of glucose and exercise. So at any one time, I'm going to talk about the average adult individual. Let's just assume that the total blood volume is 5 liters. So now we're going to figure out, if we all stood up right now to start exercising, how much glucose would we have immediately available for our cellular requirements for energy production. So if your total blood volume was 5 liters, and we know that in general, for someone who doesn't have a glucose disorder or too high or too low, the average kind of normal blood glucose is 100 milligrams per deciliter. So let's just say normal average Joe, 5 liters of blood volume, 100 milligram per deciliter. That equates to about 5 grams of glucose in our blood. And if we know that the kcal requirement of 1 gram of carbohydrate is 4 kilocalories, we can figure out that the amount of glucose circulating in our blood can provide about 20 kcals of energy. Is that a lot of energy? Not a ton. What kind of activities could you do that would allow you to expend, or how quickly would you expend 20 kcals of energy, do you think? What about during a max test? How quickly do you think that would go away? What? What'd you say? Yeah, maybe like a couple minutes, right? 1 or 2 minutes. So I don't know. I don't think we have very many pure aerobic athletes in this class. But like if you're running like a 6 miles per hour on a treadmill, a couple minutes, and you've already burnt 20 calories. So it's not that much. So this is what you have immediately available. So you need to have all those other hormone interactions to provide additional glucose. Because even 100 milligram, that's normal level, but that's not going to sustain you more than a couple minutes. So you're going to need the action of glucagon. You're going to need the action of epinephrine, in order to provide the glucose necessary for you to continue exercising. So when you start exercising, you need obviously more glucose. So we're going to need to increase the amount of glucose from our gut. So assuming you had some sort of leftover carbohydrates from your meal, we're going to need to increase the absorption from the gut to allow that glucose to go into our circulation. We're going to need to break down and free up glucose from our liver. And also our kidneys are going to help with some conversion as well. These different units can release what we call glucosyl units. And so this is, I mentioned, the precursors. So the predominant precursor that we've talked about in class, but you're going to use the most of, is lactate. As you're exercising, you're producing lactate. We can use this lactate precursor to then help us reform glucose or form our acetyl-CoA to go through our Krebs cycle and use that for aerobic energy. What we do see during exercise is that glucose, or as insulin goes down, you can see that our level of glucose rises initially and then plateaus. That's typically because we have more glucagon being secreted. So insulin is going down because insulin does what? What does insulin do? It drops blood sugar, right? So it's going to say, hey, we have plenty of glucose, let's pull it in so we can use it for our working muscles, right? So it is involved in pulling in glucose for those working muscles. But we also know that during exercise, we don't necessarily need insulin-mediated glucose uptake, right? Exercise will do that on its own. So we do see a decrease in insulin because of that antagonistic effect of those catecholamines and glucagon. So we know what insulin promotes. We also know that in the absence of glucose in the gut, so say you exercised, let's say you ate dinner at like 5 or 6 at night, then you slept, you were lucky and you got to sleep like 10 hours, right? Nice 10-hour sleep. You go to the gym fasted, so you haven't eaten anything yet. So let's just say you don't have any glucose in your gut. So very little, maybe your resting blood glucose might be a little bit less than 100. So you have even less available for energy. So we're gonna need something that's going to trigger more blood glucose. So you're going to have potentially an increased stress response, right? Increased norepinephrine, increased epinephrine, and we're going to see some shunting of the blood. So this is in part feeds into that feed-forward mechanism because with that blood shunting that occurs during exercise, we're shunting blood to our working muscles. We're also going to see some diversion of glucose to the working muscles. So blood glucose will be preferentially sent to the muscles involved in that exercise in order to spare any extra glucose you might have to provide energy for the brain. So we see release during exercise by these various tissues that have glycogen or glucose stored, and we're going to see our glucose-mediated translocation transporters, GLUT1 and GLUT4, activated in the skeletal muscles. And so we've already talked about at least GLUT4 a little bit. So we're going to see GLUT1 and GLUT4, typically more activation of GLUT4, and we see this translocation of GLUT4 via that insulin-mediated second messenger system, right? So insulin is going to trigger that cyclic AMP cascade. That will then trigger the upregulation and translocation of GLUT4 to the cell membrane, and that's going to allow for glucose to be transported into the cell. What we do know from, I wouldn't say a preponderance of studies, but there have been quite a few studies that have investigated the insulin-like effect that exercise has and the ability to pull glucose in. And the generally accepted mechanism is that these GLUT4 translocation proteins are located very closely or in proximity to the terminal cisternae and the sarcoplasmic reticulum within the cell. So that when we begin exercise and we have the release of that calcium to contribute to force production, that calcium may trigger the translocation of GLUT4. And so that seems to be the kind of generally accepted idea, but they're still doing, and I say they, as in the collective researchers of the world, are still doing some research to look into the specific actions. But from the initial studies, it seems like that it's the calcium release from muscle contraction that is helping with the translocation of GLUT4, and that's why exercise can be used as a form of medicine for people who are type 2 diabetic, because they don't necessarily need insulin to get the glucose into their working muscles. I have another link for you guys. Again, I don't want to violate copyright laws, so it's just to show you the mechanism of action of the insulin receptor, just because I think it's kind of neat. I'm not going to test you on this or anything, but if you would want to look at the insulin receptor in a little bit more detail, I've provided a little video link for you. So I mentioned that glucagon is an antagonist to insulin. Epinephrine and norepinephrine and cortisol are also known as antagonists. And again, just think about what they do. And they work in opposition of each other. So if insulin is going to try to decrease blood glucose, it's going to try to pull glucose into cells, it's going to increase protein anabolism, increase the creation of triglycerides. These are involved in producing energy, right? So it's trying to make energy available to use. And so that's going to be the opposite of what insulin does. So our two catecholamines, and then we've discussed cortisol as well. What other hormones are involved in carbohydrate metabolism? I teased a little bit about growth hormone. So growth hormone is involved predominantly because it increases free fatty acid mobilization, and that can in turn affect glucose metabolism. So theoretically, if you had more free fatty acids in circulation, you'd have more free fatty acids available to use for energy. And then you could spare a little bit your glucose. However, if you had too much or over-secretion of growth hormone, then that can lead to glucose homeostatic disruptions as well. And that's what you see with people with giganticism, like Andre the Giant. T3 and T4 are also involved because they increase glucose secretion or mobilization and fat catabolism or fat mobilization. So again, more energy available from other sources if you have the secretion of those other hormones, which, lucky enough, are secreted in greater quantities with exercise. So remember, exercise is going to increase all anterior pituitary hormones. That's growth hormone and TSH. TSH is going to trigger the secretion of T3 and T4 from the thyroid. This is a little summary of our discussion on carbohydrate metabolism. So again, exercise intensity and duration. Just like if you have done an exercise prescription class, exercise intensity is like the king of all variables, and it does have the greatest effect on the secretion and the amounts of secretion of these hormones. So the intensity of the exercise definitely will influence how much of growth hormone, how much of TSH you're secreting. Typically in very low intensity exercise and like submaximal steady state exercise, you're not going to see a significant change in growth hormone or TSH or T3 and T4. If we want to look across these hormones across time, I think it's kind of neat just to see how even though they might have similar actions or similar goals in terms of increasing energy, we can see that the amounts of the hormones will definitely vary as a function of time and also how much they change over time. So the purple is cortisol. So we can see cortisol is spiking about 30 minutes. And then assuming like a normal healthy individual who's not like overly stressed or anxious, we see a steady decline across time. If we look at glucose, glucose is being maintained. So glucose is being maintained because of the actions of these other hormones. Glucagon is initially going to spike at the beginning of exercise, and you'll see that it levels out. And then we have a steady increase in epinephrine and norepinephrine across time.
Is that 3
hours? Yeah.
So
I'll have to get the study for that. No. This would be like a marathon. Like a marathon. So we've talked about that feed-forward mechanism. So again, remember specifically it's having to do with the catecholamines. And the catecholamines are, although they release glucose from many different sites, it's the specific discussion of how they stimulate the glucose secretion from the liver. So breaking down glycogen stored at the liver will help with that immediate or fairly immediate need for energy at the beginning of exercise. So at the very beginning of class, we talked about the negative feedback loop with extracellular blood glucose concentration. This is the alternate hypothesis, and this is specific for exercise. So at rest, the general accepted theory is that it's negative feedback. During exercise, there is some negative feedback at play, but in order to compensate for that need of energy, because you only have about 20 kcal available, we need this feed-forward mechanism. So let's talk a little bit about fat metabolism. Again, we've already talked about a couple hormones that will increase free fatty acid mobilization, but it's really important for endurance performance. As you become more higher levels of endurance training are also going to allow you to better utilize your stored fat for energy, and so we're going to see that come into play with our hormones as well. So lipolysis, the breakdown of fat, is decreased or stimulated by the decrease of insulin. Remember, increased insulin is telling us what? What does having a lot of insulin tell us? Have a lot of blood glucose, so we don't really need energy, right? So if you have less insulin secreted or in circulation, that probably means you need some energy, right? Because you don't have a lot of surplus. So that would then stimulate the breakdown of fat. Epinephrine, norepinephrine, those are catecholamines, fight or flight, free fatty acid mobilization. Cortisol, stress hormone, more longer term, needs some free fatty acid, and growth hormone is going to do that as well. And that's going to work with enzymes termed lipases. So lipases are going to help break down stored triglyceride. So in general, training, and these are some training adaptations and responses, training in general reduces some of the hormone responses to glucagon and the catecholamines, because you don't need as much, because you're becoming more efficient, and typically you're going to be able to utilize fat a little bit more efficiently for exercise, especially during submaximal exercise. And again, this kind of makes sense if you think about a training response. If you train at the anaerobic threshold or above the anaerobic threshold for a few weeks, 3 or 4 weeks, then if you start at the intensity that was once your anaerobic threshold at the beginning of those 3 or 4 weeks, now that is a theoretically lower overall intensity. So you should be consuming less oxygen. It's going to put less strain on glucose metabolism and fat metabolism. So that would become your new kind of submax. We're also going to see a temporary or attenuated reduction in insulin. So insulin will decrease a little bit, but it's going to offset that decrease. So it's going to allow insulin to be secreted and maintain circulation within your blood plasma for a little bit longer, which is going to make you more efficient at pulling in glucose or oh my gosh, just lost me. Pulling in more glucose or stimulating protein metabolism. So anabolic response associated with circulating amino acids. We also see, and we've talked a little bit about mitochondrial adaptations earlier in the class, and I don't know if I mentioned the GLUT4 adaptations in a previous section, but we do see exercise training increases our quantity of GLUT4 receptors in proteins and increases the stimulus for their translocation. So it's going to be easier for them to reach the cell membrane to allow glucose to flow in. Training responses will also vary depending on what percent of VO2 max. So typically the higher intensity is going to be associated with a greater response, and there haven't been any significant changes demonstrated below 50% of VO2. So if you want to see a hormone adaptation associated with aerobic training, then it typically has to be greater than 50%. So this would be like moderate to vigorous in terms of categorization. So at 50 to 60%, you're going to see a modest increase in your catecholamine levels, 60%, a large increase, so more so than you would see at a moderate intensity. And then over 60%, we're going to see that trained individuals are going to have much higher levels of catecholamine than untrained. So it's going to be, they're more efficient at mobilizing the energy, both fat, protein, and carbohydrate. Okay, so let's talk a little bit about fluid regulation now. So jumping away from carbohydrate or fat metabolism. And if you've taken environmental, this should, I'm assuming some of this should be a review, but we're going to talk about it a little bit anyway. So what happens to plasma volume during exercise? So not necessarily just during the heat, but just during exercise, what happens to plasma volume? So yeah, plasma volume decreases, and so what does that cause downstream? So if we think of, if plasma volume goes down, we have less pressure within our blood vessels, within our arteries. What might that do to our osmotic pressure? Do you guys remember learning about osmotic pressure? So osmotic pressure is like a pushing, so we have filtration forces, we have pushing forces and pulling forces. So these are typically going to be based on where you have your solutes. So if you have a lot of like sodium or potassium, that's going to have more pulling power. So it's going to draw fluid towards it. If you have less of those solutes, then you're going to have more of a pushing power. So let's say we have a decrease in plasma volume. So plasma volume goes down, but there's no change in our hematocrit or our blood cell count. So think of the blood vessel. So plasma volume goes down, but the cells remain the same. Are we going to have more pressure drawing fluid in to our vessels or pushing out towards the cell or the periphery? What do you guys think?
Wouldn't you have a higher, now you're having like a higher grade ratio of solving solvent?
Yeah, so it's more concentrated
versus like filtrated plasma volume.
Yeah, so it's a hemo concentration. Typically, you're gonna have more red blood cells per plasma because you have a general decrease in plasma. So if you think of a pulling force, we're gonna have more pulling draw kind of into the vessels of the arteries than pushing out towards the periphery. So that's a little bit about osmotic pressure. So water is going to be drawn if we have a lot of cells in our bloodstream filtering past our organs, right? So we have this hemoconcentration, right? A lot of red blood cells, and then we have our organs, right? So we're pulling, now we have pulling pressure. So our vessels are going to be pulling some of this fluid from the periphery because you have all those solutes within your bloodstream. So, but where is the periphery? Where are they pulling it from? Yeah, and from the surrounding organs, right? So it's pulling from the organs basically that it's perfusing. So we're going to decrease some of our total body water even further because of this decrease in plasma volume. Now this isn't going to be as significant if you're in a thermoneutral environment compared to the heat, but it is going to happen, and it's going to be exacerbated if you're sweating, right? So if you're sweating, you're going to lose that plasma volume even quicker, and that's going to cause that change in gradient between those pulling and pushing forces, and it's going to draw more fluid from your tissues basically to try to make up for that loss in plasma volume. So we do have some hormones that will try to work to help correct these fluid imbalances. And the glands that we're going to discuss related to these hormones are the posterior pituitary, the adrenal cortex again, and then we're going to talk about the kidney as well, because these have impacts on not only your plasma volume and plasma water, but your heart function and your blood pressure. So jumping back up to the top here, we have our hypothalamus that connects to our anterior and posterior pituitaries. So like I mentioned before, we have different types of connections in terms of networks between our hypothalamus and our pituitary gland. The posterior pituitary is going to have a greater neurosecretive connection than blood flow secretion. So it's going to have neural factors. Because of that, we can't really create anything in our posterior pituitary, we're just storing it. So oxytocin and antidiuretic hormone are actually synthesized in our hypothalamus, and then they're transferred for storage, basically, at our posterior pituitary. Oxytocin that's involved with uterine contractions and mating responses and how connected you feel. So we're not going to talk about that in related to exercise, although it is interesting. But antidiuretic hormone is the hormone that we're more interested in. And it's also referred to as vasopressin. They mean about the same thing. And antidiuretic hormone is going to work to retain water. So it's going to work to increase water absorption or retention, and it acts on the kidneys. So instead of the kidneys working to filter out excess water, it's going to tell those kidneys to hold on to as much as it can to try to equalize the solute to solvent ratio. So antidiuretic hormone is predominantly triggered in response to a signal from osmoreceptors, and those osmoreceptors are going to measure osmolality. So osmolality is a measure of your concentration. So again, that solute to solvent. Hemoconcentration refers to a greater quantity of solute to solvent, and in this case, it's red blood cells. So if you have a hemoconcentration that's more cells per plasma fluid volume, then would be typically normal. And I'll show you in the next slide, we have a nice little spectrum showing the difference. If your osmolality, so this is talking about, let's talk about just the vessels themselves. So if that osmolality goes up, that's going to cause water to be drawn in because, again, water is going to be attracted to solute. So let's talk about sodium. So that's going to cause the water to be drawn in. If you have a decrease in osmolality, that's going to be a pushing force. So it's going to cause water to be filtered or pushed out. Right, so we have the pulling force, drawing it in, and the pushing or filtration force, pulling it out. So here's kind of the spectrum that those osmoreceptors can sense. They can sense a hemodilution, so that's too much plasma. They can sense normal, and then they can sense hemoconcentration. It's in the state of hemoconcentration that we're going to see the stimuli for antidiuretic hormone. So if we have this hemoconcentration, that's typically because our plasma volume has gone down. So maybe you're sweating a lot, maybe you're hypohydrated, you don't have a lot of fluid volume. That's going to cause an increase in osmolality. That will trigger our osmoreceptors. The osmoreceptors will then signal to the hypothalamus. Hypothalamus will then secrete or drop down antidiuretic hormone to the posterior pituitary. Posterior pituitary is going to secrete antidiuretic hormone. It's way up here, but it has to go way down here to the kidney. It's going to act on the kidney and say, you need to retain or refilter some of that water. So it's going to tell the kidney directly, don't filter out all that water, please reabsorb it. Don't excrete it in the urine. So typically we're also going to see really concentrated urine if we have a higher level of ADH because it's trying to retain as much water as it can within the body. Again, I have another little animation that you can look at. It just replays what I just mentioned. So we have a soccer player here who's sweating, and the ADH is going to go then act on her kidneys to refilter that water. Who remembers learning about ADH in another? I know you do, Sergio. You do too, Zach. Which class? For anatomy phys? Okay, cool. Cool. So it is a partial review then.
I do have a question.
Yeah.
Before we get too far into it. So there's this thing that we often discuss about astronauts taking in high solute salt solutions in space to maintain their plasma volume levels. And then, like, I'm pretty sure they apply that to exercise, right? You can take.
Well, you can, yeah. So depending on what what environment or how long you might be exercising, it's not. I don't know. Maybe Dr. Moore can give it a little insight into the like salt tablet usage, because I'm not familiar with how widely established that still is. I knew that used to be a much more common practice. Like old school kind of football, they'd have the athletes when they're doing their 2-a-days swallow the salt pill. But from my understanding, it's not as common.
Because it has been isotonic solution, right? So that it prevents any. Yeah.
So you're trying to maintain isotonic, so you're not either hypo or hypertonic, because that is going to cause either cell swelling or shrinking. In space, because of the zero gravity environment, we do see a lot of fluid shifting. So we see fluid shifting from being generally distributed to we see swelling in the thoracic region in the head. So it's I've actually heard of a NASA pilot refer to it as fathead bird leg syndrome, because you have all that fluid shift. But I am not familiar enough with space travel use of salt to give you a better answer, I guess. Robert.
Dr. Jackson brought some goo. It was the right stuff. Yeah,
it's all the right
stuff. And it's literally like just salt. It's like
salt.
2,000 milligrams.
Yeah.
And the funny thing was, she didn't tell us. She didn't tell us that you were supposed to mix it in water. She gave us a sample, and so I just opened it and just like like a carb. Like I thought it was.
So it was not the right stuff then. I know during like ultra endurance races, some athletes will, if they're cramping really bad or having a lot of fluid shifting issues, they'll drink pickle juice too.
Right. She told us it was for the exact same reason.
Yeah.
Fluid shift. That's what they take.
Yeah.
But even she said it's not perfect yet, but they're trying.
Yeah. You'll even see if like the twin astronauts, I think they're like kind of have a big head to begin with, but when you return, like, so I know that they just sent out 3 astronauts to the space station. So I don't know how long they're going to be up there. But one of them is an American. So when they come back, I don't know if they even televise NASA stuff anymore, but usually the head will still look kind of swollen, like the head and the neck, when they come back from space because of that fluid shift. Yeah, it's crazy. But anyway, so back to ADH. This should be a little bit review if you've learned about ADH already. If not, then I think this is a really nice flow chart to show you how hypohydration, or the process of becoming dehydrated or being dehydrated, can affect fluid volume and how that will trigger ADH. So we just can quickly go through this. Let's say you're exercising, you're sweating a lot, you're not drinking enough, so you're going to have a decrease in extracellular fluid volume. So we're starting right. That's going to cause triggering of our stress receptors. So our baroreceptors in your heart are going to sense that decrease in extracellular fluid volume. So not only do we have osmoreceptors that I mentioned already, but these baroreceptors will sense changes in fluid shift, which can also trigger the release of ADH. So that's going to trigger ADH release. That's going to cause an increase in plasma ADH. It's going to go all the way to the kidney to work on the collecting duct, increase water reabsorption, decrease water excretion, bring you back to normal fluid volume. We also have, if we looked from what we mentioned already, the increase in plasma osmolality. That's going to act on those osmoreceptors. Again, send us back up to the posterior pituitary to secrete ADH. Osmoreceptors are also going to trigger our thirst response. However, if you're starting to feel thirsty, that's because you're already hypohydrated, right? So that means that you're already a little bit, not too far gone, but the thirst response is not sufficient to prevent hypohydration. Another hormone related to fluid balance control is aldosterone. So this brings us back to our little pyramid glands on top of the kidney. So we've zoomed in again onto the cortex, but now we're in that first layer. The zona glomerulosa is going to secrete our mineral corticoid, and the primary one is aldosterone. Aldosterone has a few different stimuli, but it's going to work to retain sodium. So ADH is retaining water. Aldosterone is working to retain sodium. Some of the triggering responses: a decrease in plasma sodium, a decrease in plasma volume, decrease in blood pressure, or an increase in plasma potassium can all trigger the secretion of aldosterone. So one great thing about the human body is there's a lot of redundancies built into the system. So if any one trigger or factor should fail, there's usually multiple other factors that can take its place, and they're usually interrelated. So for example, if you have a decrease in plasma sodium, water typically follows sodium, so it's also very likely that you're going to have a decrease in plasma volume or fluid volume. We know that there's appropriate ratios of sodium and potassium, so if your body senses that the concentration of potassium is relatively high, that probably means that the concentration of sodium is low. So even if it doesn't sense sodium outright, it can sense changes in potassium as well, and you'll get the same response. What we see is that aldosterone is going to be triggered in part from the kidneys, but then it's going to come back and act on the kidneys. So the kidneys will sense changes in sodium concentration, changes in plasma volume, and that will then trigger a response using the enzyme renin that can then help you secrete and create aldosterone. So let's look at this. I'm going to kind of go here, and then I'm going to go back.
Was EPO erythropoietin?
Yeah. We don't have time to talk about EPO. It's so cool. EPO is involved in red blood cell production, and it's used by some to help with blood doping. But it's really interesting. So that is also secreted from the kidneys. But so we're going to focus on ADH and renin. So when our kidneys sense a decrease in plasma flow or a decrease in sodium concentration, that's going to release an enzyme called renin into the blood. We're also going to have the simultaneous release from our liver to secrete angiotensinogen. These two are going to meet up in the bloodstream. So angiotensinogen is going to meet up with renin, and that's going to convert angiotensinogen into angiotensin 1. Angiotensin 1 is going to circulate through the bloodstream and head towards the lungs. Funny enough, so you're starting at the kidneys and the liver, meeting up, traveling all the way back through systemic circulation to your lungs, and that's where we're going to see the enzyme angiotensin converting enzyme, or ACE, convert angiotensin 1 to angiotensin 2. Angiotensin 2 will then re-enter circulation. It's going to act on our adrenal cortex at the zona glomerulosa, so that top layer. That's going to secrete aldosterone. Aldosterone is going to circulate back to the kidney, and it's going to tell the kidney to retain sodium. So it's going to retain more sodium, thereby increasing how much water is retained, right? So water follows sodium, so the more sodium you can retain, theoretically, the more water you will also retain, and that can help increase our blood pressure or maintain our blood pressure and equalize that decrease in plasma volume. Has anyone heard of ACE before, or in common? Sergio, where have you heard of ACE before, Sergio? Well, what about, so on TV, right? They usually have way too many drug commercials than we'd probably need to see, right? An ACE inhibitor, right? So you guys have, most of you heard of an ACE inhibitor? Okay. So a lot of times people will be prescribed ACE inhibitors if they have issues with blood pressure, so hypertension. Because let's look at what ACE does, right? So ACE is going to help us convert into angiotensin 2, which is going to act on aldosterone. Aldosterone is going to retain sodium. Then what's sodium going to work to do? So if we're retaining sodium, what else are we going to retain? Water. What is that retention in sodium and water going to do? It's going to increase blood pressure. Ideally, make it normal in a normal person. So, but if you are someone with high blood pressure, then you're trying to find any way you can to reduce blood pressure, right? So ACE is contributing to that aldosterone-mediated rise in blood pressure. Now, for someone who's not exercising, that's fine. You know, if you have high blood pressure, you could be on an ACE inhibitor. What if that person's exercising in the heat and they're sweating a lot? It might put them more at risk of heat illness, right? So it might put them more at risk of heat stress and heat illness because they don't have this other protective mechanism. So for like everyday, normal situations, it's okay, but this is also another reason why you go through a health history questionnaire and informed consent and stuff when you're having someone be a participant in any research study, but also any exercise program, so you can be aware of these additional strains and stresses that they might be at risk for. So another animation you can take a look at, just going through that flow chart of renin, angiotensinogen, all the way back to aldosterone. So ideally, if you're retaining water, you're retaining sodium, you can increase the chance that you can retain a normal plasma volume and maintain blood pressure. And again, that's all as a response of either a decrease in plasma volume or that hemo concentration, that increase in osmolality. ADH and aldosterone, the effects are fairly long lasting though. So typically you're gonna have, feel maybe a little bit puffy or extra hydrated if you end up having these two hormones triggered. So if you are, say, hypohydrated and you have these hormones being released, maybe you're going to be consuming some sodium or consuming more water. These hormones don't just disappear. You know, they start that triggering mechanism, and so sometimes people will feel a little bit swollen or puffy, or maybe have some slight edema after the fact, so after a long race or exercise activity, because of the long-lasting effects of these hormones. And I've included a little summary. Again, we only talked about really 2 steroid-based hormones today because we skipped the reproductive hormones, but this is just a nice categorization of if they're peptide, amine, or steroid-based hormones. And there's a lot of other really cool hormones that we don't have time to talk about, but I encourage you to take an endo class if you have the opportunity to do so. Any questions before we stop for the day? No.
Are we expected to know where each one of these hormones is secreted from?
I would say that although, you know, I'm standing up here talking, I don't particularly like